the old saying "you are what you eat" has gained a new meaning. Our research reveals that it is the gut microbiota that truly determines whether dietary phytochemicals can exert their health benefits. Join us on this scientific journey from "you are what you eat" to "you are what your microbes make of what you eat." 3 key highlights. First, uncovering the microbial origin of the phytochemical efficacy paradox. Second, establishing a research paradigm of omics mining plus experimental validation. Third, providing scientific foundations for next-generation probiotics, postbiotics, and personalized nutrition. Why do phytochemicals show strong effects in lab and animal studies, yet their benefits vary greatly among humans, sometimes even failing? This is the efficacy paradox. The reason is that up to 90% of dietary phytochemicals cannot be directly absorbed. They reach the colon where gut microbes metabolize and activate them. Thus, the true determinant of health is not just what you eat, but what your microbes make of what you eat. So, how exactly do microbes do this? Take ellagitannins as an example. Found in pomegranates and berries, they are poorly absorbed. In the gut, specific bacteria stepwise convert them into urolithins. Urolithin A has antioxidant and muscle-enhancing benefits, but only some individuals, metabotypes A or B, can produce it. Key converting bacteria include Gordonobacter and Enterocloster, whose dehydroxylase genes determine whether you benefit. A similar conversion logic applies to soy isoflavones. The health benefits of soy isoflavones depend on whether they are converted to S-equol, a potent phytoestrogen. Only bacteria carrying specific gene clusters, e.g. Schlackea, can perform this conversion. Likewise, flavin-3-ols from tea and cocoa undergo multi-step microbial metabolism to produce phenylgamma-valorolactones, which exert anti-inflammatory and blood pressure-lowering effects. These interindividual metabolic differences are the direct cause of the efficacy paradox. So how can we systematically study these microbial transformations? To move from correlation to causation, we integrate multiomics, metagenomics, metabolomics, metatranscriptomics, and bioinformatics to rapidly screen candidate strains and genes, followed by classical experiments such as strain isolation, gene knockout, and enzyme assays to validate function. This discovery-to-validation pipeline has successfully identified urolithin dehydroxylases and S-equol biosynthesis gene clusters. Based on these mechanisms, what practical applications can we develop? The first application is next-generation probiotics, NGPs, well-characterized strains from the healthy human gut that can compensate for missing metabolic functions. For example, Schlackea isoflavona convertans converts daidzein from soy into the potent phytoestrogen S-equol. NGPs have clear mechanisms of action, and their efficacy can be measured by biomarkers such as S-equol levels. However, using NGPs alone still faces challenges, prompting us to develop more refined combination strategies. The second application is personalized symbiotics and precision prebiotics. Symbiotics combine a specialist converter with its preferred substrate, for example, supplementing S-equol non-producers with Adler-Creutzfeldt equalifications together with soy isoflavones to help them produce S-equol. The third application is postbiotics, direct bioactive metabolites. Urolithin A improved muscle health in trials. S-equol and PVLs are promising. The fourth application is precision fermentation, convert phytochemicals during food processing. Add S-equol producing strains to yogurt, or engineer S. cerevisiae or E. coli. Benefits are consistent for everyone. These 4 strategies point to one future. In summary, the variable efficacy of phytochemicals in humans originates from differences in gut microbial metabolism. By integrating multiomics with classical biochemistry, we are mapping the microbial metabolic pathways underlying the health benefits of our diet. Resolving the efficacy paradox will pave the way for a new era of personalized nutrition, allowing us to precisely tailor diets, supplements, and functional foods to each person's unique microbial makeup, ultimately unlocking the profound health potential hidden in food. The future of functional ingredients lies not merely in the plant itself, but more importantly in the potent molecules created by our gut microbiome.